Hello, everyone, and thanks for attending my NC Space Grant Lightning Talk. I'll be talking about the circadian regulation of the Arabidopsis gravitropic response. The circadian clock is a pervasive regulator of an organism's physiology. In Arabidopsis, virtually the entire transcriptome shows some rhythmic gene expression, while 20 to 25% of the transcriptome are direct targets of the core circadian clock. One important type of circadian regulation is circadian gating. What circadian gating means is that during two different times of day, the same stimulus given to an organism will result in two different responses. During the non-permissive time, there's no physiological response, but during the permissive time, there is a robust response. That stimulus gets through and the plant responds. In order to evaluate whether or not the circadian clock plays a role in the gravitropic response, we decided to do a classic root bending assay over a 24-hour time course, turning the plate every 2 hours to look to see if there was any difference in how the plants respond. We also repeated the experiment in constant conditions after the same period of entrainment under 12-12 lights. And this is done to better determine whether or not the circadian clock is truly involved or some other timekeeping mechanism. We used Raspberry Pi cameras to image our plants, and these were the types of images that we were getting. One initial piece of data that we wanted to check is to ensure that turning the plate itself wasn't altering the total amount of growth that a plant was doing during the time course. And we found that the total amount of growth done over the 24-hour time course was the same for all time points. What we did identify, though, is time of day dependent variation in the root angle. So I'm just showing some representative examples of our two most different time points, ZT12, which is dusk, and ZT22, which is 2 hours before dawn. But we did see variation throughout the time course. What we wanted to do is select these two time points highlighted in red for closer evaluation. So what we did first was just explore what the root angle was doing throughout the 24-hour time course. And what you might notice here is that ZT22 jumps out to this early lead in the root bending race, but then ZT12 eventually surpasses ZT22. So we thought that was sort of interesting. And we wanted to look more closely at the root growth rate throughout the time course. What became immediately clear is, as we expected, ZT22 was peaking in its growth rate. This has been established before, that root growth rate increases through the night and decreases through the day, while alternatively, ZT2012 was at its trough of growth rate right as the plant was being turned. So if we adjust the root bending angle by the growth rate, those differences are maintained for the root bending. Like I said, we repeated this experiment in constant conditions and identified similar results. We also evaluated root growth on microgravity simulators, first using clock mutants on a clinostat, a 2D clinostat. Perhaps most interestingly is the CCA1 overexpressor mutant, which is locked in a sort of permanent morning state, shows very different response to clino-rotation depending on whether or not it's grown under 12-12 light conditions or constant light conditions. We utilized the same two most different time points, ZT12 and ZT22, on the RPM, and identified some gross differences in how much reorientation and bending they were doing on the RPM. But because of growing in and out of the plate and some other issues, these were hard to identify statistically. So that's all to say, we think it's really important to consider the clock when doing your root gravitropism experiments. We think the root gravitropic response is gated by the circadian clock, and that makes some sense. Things like auxin, starch, and root growth, we know are regulated by the circadian clock and are important players in the gravitropic response. So I just want to acknowledge my funding, NC Space Grant and NASA, as well as my co-advisors, Dr. Doherty and Dr. Pereira, as well as the rest of my committee and lab, folks at the Kennedy Space Center, and the members, the undergraduates who did the prior work. Feel free to email me at jsjoseph ncsu.edu if you have any questions or would like to talk about things in more detail. Thank you.